All right, this is part 2 of our lecture on the Intro to Biology. And last time we talked about the 3 domains of living things. And recall that the archaea are the simplest of organisms. They are prokaryotes, which means they lack a nucleus, and organelles, membrane-bound organelles. And these live in extreme environments, such as salty environments, extremely hot environments like the hot springs, or very acidic environments. So these are not as popular as the most common prokaryote, which are the bacteria. Bacteria are very numerous. They're all over. They're inside our body. They're on the surface of our body. They surround us. Luckily, we have an immune system that helps prevent infection in most cases, but we do know that they also can cause infection. And some of us have probably had bacterial infections such as strep throat or a staph infection or urinary tract infection. So we know that these are everywhere, and it's just a matter of keeping them in check to make sure that we don't take them in our food or become exposed to them in a healthcare environment. The eukarya is the largest domain, contains all of the organisms. Some are single-celled, like this paramecium. Others are multicellular, but they are eukaryotes. We call them eukarya because they do contain a nucleus and membrane-bound organelles. So some concepts related to biology then are, one is the concept of natural selection. We know that organisms evolve over time and specific traits are selected for within the environment. For example, we know that a certain plant is preferred to be eaten by a deer, but the deer only prefer the plant that have the smooth leaves. So if over time a change in DNA, a mutation, causes that leaf to become hairy, the deer will avoid eating that leaf. And as a result, more of those leaves will be found in the environment because they're not getting eaten by the deer, and then they're able to reproduce, and we see a majority of those types of leaves then occurring in that plant. So it starts with a mutation, but then those traits are selected because they survive more because of the trait, and as a result they increase in frequency, and we see this change over time, which is promoted by this process of natural selection, where favorable traits are selected in the environment because they promote their survival and their ability to reproduce. Another concept in biology is that chemicals flow through a cycle. So we know that the sun produces energy and the plants use that energy to create glucose, to make glucose. So they convert that solar energy into chemical energy. And then when the rabbit eats that chemical energy, that glucose produced by the plant, that energy flows into the rabbit. And then if that rabbit is eaten by this hawk, that energy from the rabbit flows into the hawk. The same thing occurs with the mouse. The mouse eats the grass, and the snake eats the mouse, and the hawk could eat the snake also, and energy flows from one system into another. And we know as an animal breaks down, as it decays on the surface, the chemicals in that organism, in the cells of that organism, help provide nutrition for the soil. And we also know that animal waste is another form of nutrition for the soil. So we know that, for example, manure is a popular fertilizer for farm fields because that nutrition from what that animal ate does pass through into the manure, so that nutrition then can be absorbed back into the soil. So energy flows from one system to another, and all starting with the sun. So the sun is the source of energy here on Earth. So looking at the human population and our Earth, we know that humans have the biggest impact on the Earth's biodiversity and overall health. Because we have a need for housing and food, we have to sometimes deplete some of the important resources that maintain ecosystems. And some of that is through the forests. Major grazing land is created in third world countries by cutting down tropical forest trees. They can sell those tropical forest trees for money, and then creating that grassland for agriculture and for cows to graze, so they can sell the cows for meat. Oftentimes some of the major fast food chains are using beef from third world countries as a result of deforestation. And also we know that our activities, such as transporting goods through waterways, results in oil spills and contamination. So we are destroying some of those wetland areas, which affects the population of organisms that live in those areas. So we need our environment to maintain a healthy status because we want food that's not contaminated. We need a good abundance of food, so we need healthy soils and space to grow our food. So it really requires us to be diligent in how we impact our ecosystem if we're going to be able to get the things that we need from the earth. Again, food, certain medicines that can be derived from plants, and raw materials such as water is a big one that we need to survive. So we need to keep our waters as clean as possible because it becomes very expensive when we have to add extra cleaning processes and filtering processes to maintain a healthy water and amount of water. So here's an example of how we've impacted the coral reefs. Back in 1975, very little impact on the coral reefs. And 2004, we see significant irreversible damage to some of those coral reefs. And those coral reefs, again, impact other organisms. It's not just the one organism we're concerned about, but we're looking at all the other organisms that depend and rely on a healthy coral reef. So maintaining biodiversity is really something that should be a concern of all humans. And how we define biodiversity, it's the total number of species and their abundance relative to other species. So again, if you look at a farm field, there's not a whole lot of biodiversity there. That's what we call a monoculture. It's just one crop growing over an expansive area. So of course that's necessary for food, but we want to make sure that we're really looking at how much of the biodiversity we're converting to monoculture, and to really do some smart farming practices so we don't just strip the environment of all the biodiversity and all the impact that those different organisms have. Some of those impacts we're not even aware of, how they affect this ecosystem as a whole. And we see that organisms are being lost every day, estimating about 400 species per day due to human activities. So that's a significant number of organisms and biodiversity that's being lost. So the last part of our discussion in introducing the science of biology is knowing what science is. And science is really based on questions. We make observations in our environment and we develop questions that we want to answer through a scientific experiment. So when we look at the knowledge that's acquired from science, it's empirical, which means we are looking at information and basing it on observations and running it through tests, gathering data. Data is it's essential when we talk about knowledge that is empirical. It's based on data. It's not just based on speculation or personal opinion. So science is always changing. Well, sometimes we have one view of what we understand about a topic in science, and then that can change as we gather more information. So we'll talk about some of those as we get into the specific areas, but a real common one would be that in the early 1900s we didn't know about bacteria. We didn't know that there were these microscopic organisms causing disease. So we used some pretty crude practices to fight infections, such as bloodletting, where we thought it was bad blood that caused people to be ill, rather than trying to eliminate the disease-causing bacteria in the blood, such as with the introduction of antibiotics. So it's always changing. We're always learning. We're always testing our hypotheses. So it's not a science that has an end in mind. We're always developing and gaining more knowledge. So we base it on hypotheses, which are predictions that we can make from observing things in the environment. Some of these predictions might be incorrect. We might test it and find that it's not true. For example, if we say that a fish plus a chemical causes death, can we assume that a bird with that same chemical will also cause death? We would have to test it. We would have to run a controlled experiment to see that. So that would be a hypothesis, saying that we know that this chemical kills fish. Will it then kill birds? So one could run an experiment based on that. So this would be a hypothesis saying that if we expose birds to this chemical, then the bird will die. So a hypothesis is a prediction, it's a statement, it's an if-then statement. So the scientific method is something that everybody should be familiar with and have heard before in your other science courses, probably in high school. It starts with observations, and from those observations we make a prediction, which again is a statement. It's not a question, but it's a prediction of what we think is happening based on our observations. And then we run an experiment to test that hypothesis to see if our prediction is correct. And then we analyze that data to make a conclusion, saying that yes, our hypothesis is correct or it is incorrect. So it's a matter of testing the hypothesis to determine if we need to do more testing or change the experiment if we find that what we were anticipating didn't turn out. And then if we have enough conclusions that support the hypothesis, we can develop what we call a scientific theory. And a scientific theory is something that supports what we see in our repeated experiments, and it explains the why of what we see. So that's the difference between a law and a theory, is the theory explains why something is occurring based on our experimental observations. So some examples of a hypothesis is here. This is a prediction that someone can observe and say hair that is shaved, say a woman who shaves hair on her legs or a man who shaves hair on his face, when it grows back, it will be coarser and darker. Estrogens in sewage runoff, turning fish into hermaphrodites, which means the fish have both male and female parts. That is, again, a hypothesis, a prediction. Oops. Oh, having some technical difficulties here. Just 1 minute. Okay, so again, a hypothesis is a statement making a prediction, and when it asks what is the null hypothesis is when you run an experiment and you find there is no data to support a connection, then we say that's the null hypothesis, that hair that is shaved and grows back does not grow back coarser and darker. And estrogens in sewage runoff turn fish into hermaphrodites. That would be incorrect. There is no statistical relationship. So we would say that estrogens in sewage runoff do not impact fish and their gender, or they do not become hermaphrodites. So a null hypothesis, when something is null, you've probably heard the term null and void, which means not going to happen, not true. So that's what a null hypothesis is, which means there's no statistical relationship between the two things, which means in the first one, shaving the hair causes it to grow back differently. Not true, no relationship. And it is true that the null hypothesis in this case for hair, that when hair is shaved, it's simply cut at the surface, and therefore it doesn't have a pointed tip, so it appears coarser and darker, but it's only because it doesn't have that fine end that it would have had it been allowed to grow naturally without being cut. So whenever a person shaves a hair, they create a blunt end on the hair, and it appears coarser and darker when actually it's not. So whenever we're looking at scientific data and looking at someone's hypothesis and their conclusions, we need to be able to identify the different parts that made up that experiment to determine if it's valid. So again, every scientific experiment, if it follows a scientific method, should start with some observations which cause someone to question something in the environment, and then they're going to answer that question by making a prediction. So the first part of the scientific method is designing your hypothesis, which is a prediction or an if-then statement. An example would be, if a student studies more than 5 hours a week, then their course grade will be higher than a C. That would be an example of a hypothesis. It's not a question, it's a prediction, it's a statement. So then when we set up an experiment to test that hypothesis, we want to look at the different variables in the experiment. So an independent variable would be something that is changing in the experiment, but we're not actively changing it. For example, if we look at cell phone use with age, we would find that, or we would say that the independent variable is age because it's changing in the experiment, but we're not changing any of the persons in the experiment's age. We're changing or looking at the cell phone use. That's something that can change and that we're measuring as a result of our hypothesis. The dependent variable, if we're looking at cell phone use with age, would be the cell phone use. We would say that cell phone use, does it depend on age? And in some cases, some might say it does, but the dependent variable is what responds to the variable, or I'm sorry, responds to our experiment. It changes as a result of what we're testing. So if we're testing cell phone use, then and age, then the dependent variable would be cell phone usage. And standardized or control variables are things that we don't want to change in the experiment. We want to keep it the same for the group, so we don't have extra variables. You only want to vary one factor in an experiment. And I'll give you some examples of that. And a control group is some group in the experiment that is not going to be exposed to any change. They're going to be the test group for looking at, I shouldn't say test group, they're going to be the group that we're going to keep away from our test. For example, if we're looking at a pill that fights depression, our control group would be the group that receives a pill that does not have the medicine in it. They would just receive a pill that maybe has sugar in it and is not going to really affect depression. It's just sugar pill, a placebo we call it. So that would be the control group. The test group would be the group that's exposed to the medicines, the one that's actually going to be getting the antidepressant medication in that pill, because that's what we're trying to test. And then replication just means, can I repeat that experiment over and over and over again and get the same results? If I can, then the hypothesis is valid and it's a good experimental design. So it's important that whenever we design an experiment that we identify our independent and dependent variables, and that we control all the other variables that might come into play. And we define who our control group is and who our test group is. So an example is we have a nursery that has pea plants that are exposed to 6 hours of light, and someone wants to know if I can increase the light. Will it increase the growth rate? So they set up an experiment. One group is exposed to 6 hours of light per day, one to 7, and another to 8. So he measures and records the growth rate of the plants in each of those groups. So the hypothesis would be, well, he wants to know so we say if we increase the light exposure, the growth rate of plants will also increase. That would be a good hypothesis. The independent variable in this case would be the hours of light, because that's not something we're going to change within each group. We're going to keep one group in 8 hours, we're going to keep one group in 7 hours, and we're going to keep one group in 6 hours. So the hours of light, is something that's not changing within each group. It's staying the same. So it's the independent variable. The dependent variable in this case would be what's going to change. Well, we would expect the growth of the plants to change, so that would depend on the amount of light. So the dependent variable depends on the independent variable in this case. So the dependent variable would be the growth rate of the plants. The control group then would be the group that is exposed to less amounts of light. So if we're looking at 7 and 8, that would be higher than the typical 6. So the 7 and 8 would be the experimental group, and the group that's exposed to the normal amount of light, the 6 hours, that would be the control group. And accounting for replication, he just would want to make sure that he's keeping his variables the same, as far as the same plants, the same soil, the same amount of water, the same type of light, making sure that he can repeat that experiment and not have extra variables coming into play. So whenever we finish our data gathering, we want to look at our data and analyze it and make a conclusion. So we're either going to reject our hypothesis or we're going to support our hypothesis. And oftentimes, if something is supported, it'll enter a scientific journal and it will be shared with the scientific community. And oftentimes, scientific journals are based on different fields of study. There's journals for nursing, for physics, for biology, for earth science, chemistry, and so on. So it would go into the correct journal. And then as other scientists see that, they might attempt to duplicate the results to make sure that they agree. If it's something that is debatable or something that's very exciting, then other scientists might want to replicate those studies to determine if more study needs to occur, or if it's something that could be built upon for more improvement. Say a medicine is discovered, maybe to control blood pressure, then someone could say, well, does it also control heart rate, or does it also lower cholesterol? So there's other experiments that can come off of one experiment. So anyway, looking at then the different types of scientific information that comes out of experimentation, we have theories and laws. The evolution we know is a theory. Cell theory is where all organisms are made up of cells, and gene theory. It basically tells us why something happens. It helps to explain what we observe. So theories are statements or ideas based on evidence from experimentation. And they can be proven wrong. That's the difference between a theory and a law, because a law is something that just is observed, but we can't really explain why. We don't know why it happens, but we know it just happens. For example, the law of gravity. We know when I drop an object without friction that it falls at a rate of 9.8 meters per second that it falls. Now, can we explain why that happens? No, but we do know that it happens, and it can be repeated over and over and over again with the same results, so then it becomes a law. So think of theories as explaining the why, and they can be proven wrong with experimental data, where a law is repeatable. It cannot be proven wrong because it's just an observation that we don't necessarily know why it happened. We can just observe that it occurs. Newton's 3 laws of motion are another example. We know that we observe those. Every action, there's an equal and opposite reaction. That's Newton's 3rd law. We know that that occurs, but we don't necessarily need to know why or explain why it occurs. A law just tells you what's going to happen, and it can be repeated pretty easily. So whenever we're looking at scientific information, it's important to keep in mind where that information came from. If it came from primary literature, which means it came from a scientific journal that is reviewed by professionals in the field, we can pretty much rely on that as good data and good experimentation. But if it's coming from secondary literature, like it's a web page or it's an article written by one individual based on their observations and opinion, then we need to be a little bit more prudent in how we assess the validity of their claims, because we see a lot of that right now in the area of health. We have a lot of supplements that are being touted as better than certain medications, and that may be true, but there's a lot of bad science out there, and we need to really be careful before we adopt some of these holistic approaches, especially for people with chronic disease that we know have been managed well by certain medications or practices. We don't want to just throw those away by looking at secondary literature and reading a claim that might not be based on good science. So that concludes our discussion of the intro to biology and looking at different factors affecting biology. There is a worksheet that is posted underneath your link to this video, and I'd like you to print that off and look at the different experiments and be able to identify the dependent and independent variable and be able to define those terms. And then I'll have you discuss those when we come to class. I'll actually look at them, have you turn them in first for some points, and then we'll discuss them afterward.